Good morning, everyone. Welcome to this Audiology Planet webinar on P300, the event-related potentials. We have with us Dr. Associate Professor Mridula Sharma, who is an academic and researcher at Macquarie University. She has more than 40 papers on the topics related to the potentials as well as auditory processing disorder to her credit. Dr. Sharma has worked in New Zealand as a postdoctoral research fellow. She has an exposure to the Indian professional scenario as well. So she's got a wealth of experience, and I'm really excited about this webinar where she'll share us with her absolutely brilliant knowledge. Over to you, Mridula.
Thank you, Pankaj. I appreciate you inviting me. I'm quite excited to do this. I think when I was starting to look at this, I thought, what am I going to talk about? And then 59 slides later, here I am. So welcome to everybody who's keen on learning about what I can, I guess, share about P300, the technique, the interpretation, pitfalls, and applications. So the kind of things I'm going to talk about are listed over here. So there are 7 things. And what I'm also going to do is I'm going to interleave some of the studies and I guess some of the questions that we had and how we started to think about using P300 periodically. So hopefully you'll see my logic there. So going from there, let's talk about auditory evoked potentials, the very basics. And no, I'm not going to talk too much about it, very little. So what are auditory evoked potentials? And this is mostly a reminder, simply because I guess when we are working with evoked potentials, we are so embedded in that we tend to sometimes forget. So the first thing is that these are electric changes in the auditory pathway, and they are generated in response to sound, therefore auditory evoked. And potential and responses, they're used interchangeably. The other thing to also remind ourselves is about the classification. And oddly enough, there's no consistency. So we seem to have a preference for the choice of nomenclature. It could be latency-based. So for instance, middle latency responses are always referred as middle latency. But when it comes to late latency responses, there's the other option is cortical auditory potentials. But then when sometimes we use site of generation, so ABRs or auditory brainstem responses, we never call that, I won't say never, but we often don't refer to the brainstem responses as short latency responses. And then, of course, there's also the type of stimulus that we use to describe an auditory evoked potential, such as steady state response or auditory steady state responses. So the naming is often, it's just something people start using. When it comes to cortical auditory evoked potentials, there's another term that you often see, which is the slow vertex response, mostly because it's very late latency and that we seem to gather it from vertex. The main thing, I guess, from the background that I wanted to highlight was that there is no direct relationship between the response and the generator because we are recording the response from the scalp, far from the site of elicitation. And this is important, especially if you're thinking about and haven't used evoked potentials, but are now planning to venture into something like that. So bear in mind that we have to be really careful about what the absence of the response actually means. This is to show the time course of auditory evoked potentials. If we had to record how all along your auditory pathway the signal is being processed, then this is quite a nice way to show it, where you've got your ABRs, your MLRs, as well as the cortical responses. And that brings us to this. So I think auditory field of audiology, the good thing is that we've got so much research that's gone into evaluating the anatomical and physiological structures, but mostly physiology at various auditory pathway steps. So you've got the brainstem response, you've got your middle latency response that talks about just the subcortical regions, and then of course you have the cortical auditory evoked potentials from the cortex. But why do we often use these? Because, well, first of all, there are two main responses that are the most popularly used. There's the ABR and the cortical responses, but both of them are really known and are used for percept evaluation. So if we had a question about, well, what is it about percept, or what is it that we can evaluate about processing of a signal, then what do we do? And that's a question we were looking at when we came up with this study. So this study was to find out if English speakers can process lexical tones. This was a study done by my master's student, Alyssa, with my collaborators. And just so people know, what do we mean by lexical tones? We were specifically looking at the Mandarin tones. And the curious thing about these tones is that if you change the pitch or its trajectory across the timeline, in that case, the meaning can change. So here in the box, I've got these 4 tones, and all of them have the same CV, consonant vowel combination, except that the pitch is varying. And when that happens, so for instance, the T1, which is the high level, that can mean mum, whereas T2, which is the rising one, that can mean hemp, or a T4, which is this the falling one here, that can be a reprimand. So the point is that these are the same sounds, but the pitch is varying. So the question we asked was not about percept of these. We wanted to know, these are Mandarin tones and very familiar to Mandarin speakers, but can English speakers appreciate these as well? So there's no point using ABRs, there's no point using just the cortical responses, so what can we use? And what we've done is we've chosen T2 and T3 because that's been found to be the most difficult to differentiate. So the question we were asking is, what can we use to differentiate? And this brings me to the next topic. Well, as research would tell us, we actually do have another response, which is called the discriminative cortical auditory evoked response. Now, again, going back to that point about classification, you've got the obligatory responses, the ones I showed earlier. This is from an adult, a P1, N1, P2, which shows us that the signal has been processed at cortical level. The reason it says obligatory is because it is referring to the percept of the signal. And here, now we've got discriminative cortical auditory evoked potentials, which is telling us that the signal that we are processing is occurring at cortical level and it's in response to a discrimination task. So now we have another classification which refers to the actual task involved. So not just the latency, not just the site of generation, but the task. So what is this? So we've got 2 things. Like I said, we have obligatory, which is responses to physical properties of sensory stimuli. So say, for instance, you've got these obligatory responses, which I said are in response to percept. And if we were doing a visual task and we saw these very, I guess, statue-like storks just sitting there, to each one of the stork you would have a visual cortical auditory, visual cortical response, or a visual evoked cortical response. And if each of the stork was, say, whistling, to each of those whistles you would have a obligatory cortical response. But because background EEG, which envelopes, I guess, this auditory or visual cortical response is quite massive, often 10 times, which is in response to your muscle, everything you're seeing, everything your percept, your somatosensory. All of that needs to be extracted. So in order for us to do that, we have to present the same signal or a same stimulus at least 100 times for cortical auditory evoked potentials. And then eventually we average them and we get a response. But this is an obligatory response, which is a response to physical property of these visual stimulus or the whistles that you would hear. A discriminative potential, on the other hand, demands an oddball stimulus paradise. Same, all of those storks, one of them decided to wear a tuxedo and decided to dance. Now, if they're doing that in response to that change, you would have another response, an additional response, and that's the one we are interested in. Starr called it this particular response, he said it was a cognitive factors that might be modulating the auditory evoked potentials, which implies that there is further processing of sensory stimuli. So what kind of response do we get? So just to make it very clear, say you've got, instead of the whistles of the storks, we had these das presented 100 times. This is the kind of response you would get. There would be a P1, N1, P2, and this is in elderly, but any adult 18 years and up, this is the kind of response you would get. I chose this specifically because it shows the timeline quite nicely. And this is an average response from all those 100 das. Now, what about the discriminative potential? Now, instead of the tuxedo stork, say we have a ga sound, which is different, and we present this at a certain probability of 10%, which means if you're presenting your das 100 times or 200 times, then the proportion of that would be that you take 10% of those 200, which would be 10 of them, I think. Is that correct? No, it will be 20. That would be your gas, and you intersperse them so that you have sufficient number of das, interleave ga periodically to give you the response to that discrimination paradigm. And this is what you would get. You would get the same cortical response first, the P1, N1, P2, as you did to da, because you have to hear the ga first to be able to say, this is not da. And when you say you made that decision, you're getting this another positivity here, which is about 300. So anything between 300 to 400 millisecond, that's the response you get. Again, it's an average response. In order to get a very clean response such as this, you need at least, in adults, about 75 of these gas that are interleaved with the standard stimulus such as your da. This is just to show you when you presented that da and ga in the way we did, with lots of das and periodically gas appearing, this is the two waveforms you will get. Any clinical evoked potential equipment will be able to give you this. And one option is to just use the P300 here and measure amplitude and latency, or the cleaner way to do, the way you could do is subtract these two, so you're left only with P300. So going back to our study, remember we chose two tokens, the token, and this was a token which was spoken by a Mandarin speaker. It was ba. And then we extracted the a out of that. And we just used the token a, which was either rising or it was dipping. And this is just to show you it's across the time how the pitch varies. And remember, the question was, what paradigm should we use to see if English speakers who are not Mandarin speakers are able to differentiate this? And of course, We chose the P300. What did we do? So we had 7 Australian English listeners and we took 9 native Mandarin listeners. Actually, Alyssa has collected more data, but I haven't added that in. But that's my job for next that I have to do. Anyway, so we also recorded this response from 25, I guess, places on the head. But the ones, if we had to do on clinically and we had fewer options, I would definitely choose FZ, CZ, and PZ. If you had to choose only 2, FZ and PZ would be quite sufficient. So because it was research, we looked at 25 channels. The reference could be either ear, because we're not looking at hemispheric differences. We just want to know the P300 at the midline, which is your Z, your center of the head. Standard and frequent. Now, so standard is, or frequent stimuli is the ones that are presented more often. So remember I showed the da, da, da. We present 90% in your number of stimuli are da, and then you had periodic gas. So in this case, we had T2 and T3. Now what we did was the first time around, we made T2 the standard, so T2, 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 and then periodically we interleaved with T3. But then we also did the other way around. We reversed the paradigm or flipped the paradigm such that T3 became the standard and T2 became the deviant or the rare one that occurred for lower probability. And the probability is the other thing that's really important. So 80% of your total stimuli would be your standard and 20% are your deviant. The interstimulus interval, that is the time from the stop of your first stimulus to the start of the next stimulus, that's important too. Because P300 occurs a positivity at 300 millisecond, you need sufficient time for people to make a decision about the sound being different or not, being able to respond, because at the end of that, you get the P300. So the timing is important. My stimulus was 350 millisecond long. Actually, they were longer than that, which is why we've taken 1500. We presented binaurally, and this is the filter setting often for cortical auditory evoked potentials. The most important thing was the instructions. And there's a letter missing there. When you hear a different tone, there should be a different tone, please press the button as soon as possible. This is an important thing for you to note. We are giving them an indicator that something different is going to happen. And we are asking them to press or indicate in some way when the tone is different. What did we get? We did get, this is again, this is the subtracted stimulus, the response. So we've subtracted T3 as the deviant from T3 when it acted as standard, and here we've T2 as deviant subtracted from T2, which was standard. And these responses, as you can see, for both the groups are very similar. So the answer, did English speakers hear the difference in lexical tone? The answer is yes, they did. The reaction times told us a different story, but that's another day, and that's for another day. So going back to now coming the next topic that we want to discuss, which is P300 A and B. So P300 or P3, people use these interchangeably, but then there is P3B, and I'll come to that in a minute. It occurs in response to a listener consciously, so attentively perceiving a change in a stimulus. It could be speech, it could be tone, it could be whistles, bells, barking, environmental sounds, anything. And this is usually, as I said, it is an active or an attentive oddball paradigm. So the person has to pay attention and respond in some way, where listener is asked to consciously attend to a change in the stimulus. It is a discriminative potential, but then when you get the P300, it reflects audibility, discrimination, and a recognition, a conscious recognition of the stimulus. Now, I did say P3A, which implies that there is a P3B. I said P3B, and therefore there is a P3A. Picton in his book, and I've got a reference at the end for his book, he says that there are many dwellings in this neighborhood. So when you look within this region of 300 to 500 millisecond, there are quite a few different responses. But the ones we are going to concentrate on today, and mostly, even if you were to try this in your clinic setting, would be P3A and B. Now, what's the difference? So P3A is earlier and it's smaller. It's more prevalent at front of the scalp, and it is basically identifying, or it's an attention switching mechanism or an orienting response or an automatic detection of difference. And when I said it's earlier and smaller, by earlier, it may be around 250 millisecond, whereas P3B, which is larger, it also tends to be more consistent, and it's later because it may be closer to 300 or maybe just after 300 millisecond. It is largest at the back of the head or the parietal region rather than central CZ being your vertex. And the latency reflects the timing of discrimination as well as the recognition process. I'm assuming that everybody knows when we are looking at placing the electrodes, CZ, which is right the center and the top of the head, that's your vertex. And if you take, if you measure from your inion, which is, between your eyes, so from there, sorry, that's your nasion, to your inion, which is just the back of your head, just where your head bends from your occipital region. If you measure between those 2 points, that's giving you sort of like a whole length, and what you can do is 50% of that would be your CZ, and if you take 20% of that behind CZ, that becomes your PZ region, and 20% in front of CZ is your frontal or FZ. So if you don't have a measuring tape, I know at least in Australia, IKEA gives out these disposable measuring tapes that I tell my students not to throw, but to bring it to the class. So what's the nature of this P3A and B? So this is quite nicely given by Pollack, and there's a lot of work Pollack's done. So I'm going to be citing Picton and Pollack quite a lot. So in 2007, what they've highlighted these three paradigms. So the first one is a single stimulus, where you present the stimulus target, but that this target, there's nothing else, but the target is presented at random. Well, random, what I mean is that there is no rhythm to it. So it's not presented every 1 second or something. So when that happens, there is a less probability of the listener of determining that there's going to be a sound coming up now or a little later. That's what would elicit a P300. Second paradigm is when you take 2 stimuli, so say an S and a T, which is easy to discriminate, but the T is interspersed, and you will get a standard response, but you will also get the P300 because there's some level of discrimination required. But then if you introduce a distractor, right? Same paradigm, but this time you introduce a distractor. This distractor will give you this frontal, slightly earlier response, P3A. Now, this looks bigger over here, but if you keep adding these distractors, this P3A actually becomes smaller and P3B appears to be larger. But P3B is always more reliable. This is just to show how you would measure the amplitude from the baseline and the latency from the start of the stimulus. And that's what this is trying to, this is what I was trying to say, that amplitude is the difference between the mean baseline voltage and the largest positive peak, and latency is the time from stimulus onset to the point of maximum positive. Like I said, scalp distribution, we normally would look at, if you had the opportunity to use 3 electrodes, FZ, CZ, PZ. But if you only had to choose 2, FZ and PZ. And if you only could choose one, then you could use CZ, or if you were really sure you wanted to only look at the P3B, the parietal region, I would choose the the PZ. Mind you, the other thing also to note is that the amplitude generally does increase as you go from FZ to PZ. All right, so that tells us a brief on these two components, but let's talk about the nature. Now, this is kind of an interesting, and this is when it gets a bit interesting when we talk about why do we get a P300. So this goes back to a study by Squires, and it holds true even now. So what this is showing is the two tones here. The continuous line has a probability of, say, 10%, and the dotted line has a probability of 90%. The implication of this, the tone that is presented 90% of the time is your standard, right? So you don't really expect to see a P300 to something that's frequently occurring. And do you see that? No, of course not. What we're seeing is that the continuous line where the stimulus was presented only 10% of the total stimulus, that's when you are seeing a P300 response. But this is when the person was ignoring, right? But there is a massive change, you would get a P3A. But when you ask the person to actually attend for a difference, you get a P300 complex response that includes both the P3A, smaller, earlier, and P3B. You don't really have to try and differentiate between the two. You don't have to try and, separate these responses, because even if you do, right now we don't really understand what that means. So just appreciate the fact that you get kind of this combined response. This is the other the study, which is very useful, especially when we're talking about the nature of P300. So what this is showing here is when the P300 to a discrimination at a paradigm, an oddball paradigm, was applied with normal subject, continuous line, and people with hippocampal lesions. Now, this is a region just underneath where your temporal lobe is, and I have a picture of that later. When these people were tested, basically the people with the hippocampal lesions had no response to a novel stimulus, so that distractor at FZ. But when the PZ, where you measure the target of your oddball paradigm, when that was looked, basically there was no difference between your normal subject and hippocampal lesions. The implication of this, basically the hippocampal is involved at least in your P3A response, which is from the frontal, which is slightly earlier, and is often noted for a distracted, something completely unexpected. Okay. And something similar was also observed in another study with the frontal lesion cases, which again it indicates to us that at least two sources that are contributing to P3A are hippocampus and the frontal lobe. Now, nature of P300, so we have to talk about the theories. Now, before I actually talk about the theories, I want to highlight this part here, which came from a meta-analysis study by Teran in 2014, and this is what they said. After almost 50 years of intensive research with over 12,000 publications on P300, we still are not sure, or it has not been possible to link P300 to a specific cognitive process. So putting this aside, the two theories that are kind of try and explain why we get a P300 are these. The first is the context closure hypothesis. Now, what this is trying to say is Remember I said to you that the instruction we give to a person when we are doing a P300 is respond whenever you see or hear something different, which means that it's not unexpected. They know something different is coming. So this context closure hypothesis is stating that actually when they do hear something different, it's not a start of a suspense, but rather an end of the suspense. They were anticipating something different to occur, and it did. So that's what they're saying, that P300B is a response to a closure process, that they anticipate the deviating target stimuli, and when it appears, that's the answer to their expectation. It's a fulfillment of an expectation. And they cite a particular study where people were given, for a visual paradigm, there were a lot of pictures of models. And what they found was that the female models elicited the biggest P300, but only when the participants were males. Anyway, I thought it was just a fun story. Then the next one is the P3. The next theory is about context updating. This is when an improbable event occurs. That is, that the person's mental model of their environment or context is evaluated and updated when a relevant and a deviant stimulus is presented. That is, when you're hearing the tone, the deviant comes along, it needs to be updated to your working memory processes to then decide and respond to that difference. Now, as I said, there's no agreement on which one is a more appropriate one, but the strategic information processing, which is another, I guess it's not really another theory, but certainly been proposed that maybe what's happening is that when you get a sensory input, it goes through these top-down attention processes, and in prefrontal lobe, the decision is made that there's something novel that gives a P3A, but then it needs to update into working memory, into the memory storage, and where in temporal parietal junction, it's decided that it's different, and how different it is, that yields a P3B. So there is a top-down attention switching, as well as bottom-up memory-driven paradigm, memory-driven system that guides the response organization and production. And the frontal attention mechanism, it governs the neural responsibility to this novelty. Bottom line, the stimulus characteristic and task demands then define the topographic and timing outcomes. That is it going to be a P3A or a P3B? And therefore, the timing is going to be different, the amplitude, etc., will all change accordingly. Now, as promised, neural generators. Now, this has come from a number of intracranial studies, scalp studies, and like I said, there's the hippocampus, there's the prefrontal cortex. These are the ones that are clearly contributing to P3A. But then we also have the superior temporal sulcus and even the intra parietal sulcus pointed out here that also contribute to the P3B. And another fun fact, so for those who are in UK, it appears that UK taxi drivers, not bus drivers, taxi drivers seem to have a bigger hippocampus. And it seems to be because of their experience of driving in those very maze-like streets of London. Because there was another study that looked at it to see, was it that those with bigger hippocampus were taxi drivers? Turns out, no, it's a learned behavior. Now, there's only one study that I could find where they looked at P300 and cab drivers. And yes, indeed, it was bigger. P300 was bigger in the taxi drivers compared to those who weren't driving, but were in the same industry. Just fun thing there. Okay, coming back to what does P300B amplitude, latency, and morphology imply? Well, first of all, the P300B amplitude, it is dependent on the probability. So I've been saying that the probability often when we are testing is 80-20. The deviant is 20. Now, if you increase that probability, in that case, the P300B would actually get smaller. So the more you know the different sound is there, the smaller the P300B will be. The detection of difference is also important for us to be able to discriminate between signals of any kind. We need to be able to detect it. This detection is as important. And again, I have an example to make that point. The meaning or relevance and feedback. This is also interesting. So it appears that when you give a feedback to a person, when they responded to the discrimination, the feedback seems to drive the P300. So it's bigger when you get a feedback, and it's relatively bigger when you actually get it wrong. So that's kind of curious. P300B latency seems to be basically about completion of stimulus evaluation. And while there may be variations, and there is a lot of variations even in your normal population, the P300 morphology appears to be stable across recording sessions. So that brings us to the next point about the factors that affect P300. So first of all, there are some intrinsic factors: attention state or the arousal state. So this is what I often tell my students. If I were after an hour of listening, and that's probably what's going to happen to some of the listeners today as well, if you're tired, and that is going to contribute, and if I were recording your P300, there would probably be a difference in the P300 amplitude when you started listening to somewhere an hour later. Age is another important factor. 70-year-olds versus 20-year-olds, big difference between the amplitude and latency. And this is rather important because if you are keen on applying P300 to anything that has say a cognitive impairment, as things like schizophrenia, dementia, Alzheimer's disease, all of that, what we need is the age control. So 70-year-olds or 60-year-olds, we really need their norms. P300 latency tends to get longer. Scalp distribution is also different. So as we grow older, rather than the trajectory that I was pointing out, that the frontal FZ shows a smaller peak compared to a PZ, well, as we get older, that changes. And of course, the attention capacity. If the person is is very fidgety, they're not keen on sitting for an hour of testing, then your P300 will get impacted. There are other factors as well, and I'm going to come back to this. So paradigm, oddball. Really important that you use an oddball paradigm, and the probability of your deviant occurring is much better. So 10 to 20% works best. Inter-target interval. Now, while I said deviance, the probability of deviant has to be small, but it cannot be infinitely small. There's a limit. If the difference between two targets timing is anything between 6 to 10 seconds, they will not be a P300. 6 seconds seems to be that magic number. Again, you don't want your deviant to be rhythmic. So that's important as well. And that kind of ties in with the paradigm as well as into target time. Rate. Now, because P300 occurs at 300 and there's lots happening between up to 500, you really want a nice long time in between your stimuli to not have overlapping responses to make it difficult to read. So this is here from the Din-Terrence paper in 2014. And what they did was they wanted to gather all the papers that have looked at age effects, right? And collated that to try and figure out what is the trajectory of P300 latency and amplitude. So this box at the top is P300 latency on the y-axis and age on the x-axis. And I have kind of made it very small. I do apologize for that. This is, they managed to collate about 75 studies, and this data is from, so each of these points, there are about 1,572 participants going from 6 to 87 years of age. And what this is showing is that as you go from the 6 to about 20 years of age, the latency is shortening, which is reasonable because myelinization gets better, so neural timing decreases. But about 20 years of age, the latency starts to increase again. At the bottom in the box here, we have the P300 amplitude, and this seems to work in the opposite way, but again, it's sensible that P300 from when you're 6 years of age to about 20, the amplitude increases, but then starts to decrease again. So it appears that there is somewhere at about 20, between 20 and 25, the P300 amplitude and latency are starting to change, which is curious. But what do these mean? Once again, P300 is indexing the neural speed or brain efficiency. P300 amplitude is indexing neural power or cognitive resources, which of course increases maturation. So even if you take two stimuli and it's very easy to discriminate for a young child, it is still a lot of effort. So one of the things that P300 also seems to contribute towards is effortful or cognitive effort that requires to complete the task. I did mention about the impact of probability, and this is an old Duncan 1977 study, but it kind of makes the point very well. So what they did was is look at probability and attention in one go. So here you've got the high tone, and this is 1,000 hertz and 1,500. So 1,500 hertz is the high tone. And they changed the probability of this 1,500 hertz tone going from 10 to 90%. And what I've boxed here is basically the low probabilities. Okay, so remember that, but I'll come back to it. The task here was when you heard the 1,500 hertz or the high tone, the participants needed to count them. And the other task, in the ignore task, what happened was that they were presented with the tones, but instead of counting or paying attention to the tones, they were actually trying to work with these letters and extract words from there. So a completely different task. So reading a book could have been as good here, for instance. And first of all, if you look at these dotted or dashed line for both high tone and low tone, which is the ignore task, they're doing something completely different. Even when the probability is very high, you don't have a P300. Now, just to be very annoying, the positivity is downward. So below is so what looks like a trough is actually a peak. And your dash line is pretty much a flat line. So ignore does not work. And as the probability for when they are counting increases from 90 to 10% and actually between 30 and 10, that's when you see a P300 trough. Low tone, same thing. They just had to count the low tones. And again, so long as the probability was between 30 to 10%, P300 was present. Continuing with the story of factors, I thought this was quite appropriate to talk about. This is about detection. What here they're showing is, they are showing when you have 1,000 hertz standard presented 80% of the time, and you have 4 deviants this time. So you don't have to take 1 deviant, you can take 4. right? Which is what they did. That went from 1,025 to 50, 110, 1,500. So you can imagine that 125 hertz would be slightly different from 1,000 hertz, but then 1,500 hertz would be really different, right? These graphs here, the shaded graphs at the bottom where you've got the arrows at an angle, that's your reaction time. And can you see how that also changes as you go from low detection to very high detection of 1,500? And that's reflected in your P300 as well. So if you went, towards your J&D or just noticeable difference or close to the thresholds, you can imagine that this P300 would be really small, right? So detection is really important. Before doing a discrimination task, always good to first just test it out. Can they hear the signals to make the decision about the difference? And the feedback, the influence of feedback, as I said to you. So this is when the signal, this is between 1,000 hertz and 8,000 hertz. The probability is 65% to 35%. And of course, ignore, sure, you're still getting some responses there. But when they're counting, this is the target, you actually get a P2, and there seems to be a P300. But when they got the feedback, that's when they actually got a big response. But incomplete one got you quite a distinct responses here, which may be your A and 300A and B. But curious, isn't it, that we respond most strongly to when we get something wrong. There are, of course, lots of other factors that are equally important, but the bottom line is a shorter P300 latency and a larger P300 amplitude, and I am talking about P300B, which is from your parietal, that's associated with superior information processing. P300 is influenced by the state variables, so natural and induced, for example, gender. Females have bigger and earlier P300. I've already talked about the age factor. The body temperature makes the difference. Sleep quality makes the difference. Fatigue, exercise, food intake, drugs. There's a whole paper that's dedicated to, and I think it's Pollack 2000, where they've talked about all the factors that could contribute to the variability of P300 latency and amplitude. These are important to make when we are talking about clinical paradigms. There are all these factors that could be influencing your P300 latency and amplitude and creating the variability, which we really don't want if we want to be able to do something clinically. What I haven't got here is also alcohol or chronic alcoholism, smokers. There was at least one study done where they looked at immediate impact of smoking, and P300 did change the amplitude, changed because of that. Another thing is about this paradigm. Now, I did mention this. We did it in a study, and quite a few people do it as well. So I thought I would run past that. So the first thing is when you do your run one, say you have lots of standards, which are your DA, because they occur a number of times. And also to establish, if you think about context updating, what we also know is that you have to have DA presented at least a number first, about 10 of them have to be your standard stimuli. So you've established what is to be, and then you interleave your GA, right? So that's what this is here, where the DA is your standard, GA is your deviant, right? So you've presented GA, and you can choose your probability, anything between 10 to 20%, 30% could work as well. But then what you do is you do the second run where you make your GA the standard and DA deviant. Now, this has two advantages. The first is that you're testing both the DA and GA discrimination in both directions. When you have established DA and you hear GA, now they're different because they have differences in their formant frequencies. The second and third formant frequencies are different. But then when you have ga as the standard and you present da, do you perceive the change similarly? Some studies have shown that actually, for instance, if you have your standard as high intense stimulus and your deviant is low intense stimulus, the P300 is not as big as when your deviant is higher intensity. So there seem to be some sort of possibly adaptation or habituation that plays a role, but it's not very well understood. But nevertheless, this is, I did say two advantages. The first is about being able to check the discrimination in both directions. Okay. The second advantage is that you perceive, remember, you have to have the first response you get is an obligatory response to what you hear, right? Da and ga have different frequencies. Therefore, the cortical response to da and ga would be slightly different. So in order to ensure that the P300 peak is not influenced by the physical properties, when you extract your P300 from ga as the standard and ga as deviant, you're ensuring the stimulus response to be, I guess, extracted. And you're only left with the discrimination. Okay, so that's the second advantage. So this here is the response. See, you've got your P2 response, the rare, frequent, or deviant standard to the same stimulus. So this is minimized. However, if you take like 1000 and 1500, which are very different stimuli, your initial response would be different. So if you subtracted that, your P300 will be influenced by the responses, the obligatory responses occurring just before. So, okay. So if you needed just one slide to tell you what your protocols ought to be for P300, so you need at least two stimuli. You need a standard and a rare, or a standard and deviant, frequent or deviant, whatever be the term. Probability, 10 to 20%, no more. Have your participants seated, they're open-eyed, they shouldn't be very sleepy, so don't do it after they've had a whole day of, say, gardening and they're exhausted. I remember we talked about attention state, phasic, tonic, that's going to have an impact. Incidentally, if they're highly caffeinated, that's going to influence your P300 response as well. So detailed information, make sure you have a lot of information regarding their health and choices, which means about their alcohol consumption regularly or the day before. Are they able to sit through the paradigm, so attention to task? And like I said, the recording, I would really, and I'm not sure why I wrote CZ there, I think this should have PZ as well. And the interstimulus interval, 1 to 2 seconds. Now, P300 can be elicited by any number, any stimulus. I did show you the lexical tones we took, which were the same but different pitch. You can use a frequency. I think I've shown you examples of 1000, 1500, 1025, 1000, 8000. So you can choose anything. You can have intensity. Like I said, however, if you're using intensity, make sure your deviant has the louder intensity so that you elicit a bigger P300. You can have short versus long duration. So you can still have a short R versus a long R. You can have speech stimuli. I've shown you the da-ga. You can have a rhythmic pattern, so a complex tones, and change the rhythm. You can change it by adding a tone to it or emitting a tone. You can have the rate difference. I think I showed you an example of that. I do want to show you an example of spatial location of origin. So this is a study which is under review. This is done by my master's student, Luke, and the postdoc, my postdoc and a colleague, Vargas Peters, worked on this. This is looking at P300 as a measure of spatial localization. I think it's a really cool study. So what we did was we took all these speakers. So the person is facing in front. And from the 0 degrees, we presented the standard DAS. So most of the DAs were coming from here. But our deviant was the same DA, but they were coming from either 30 degrees, 60 degrees, or 90 degrees from the right or the left side. So it's the same stimulus, but instead what we've got is the spatial localization that's different. Now, why is this an important paradigm? Because if you're working with people with children with auditory processing disorders, this is something that is quite useful to observe. And because this has been noted to be quite different in populations with auditory processing disorders, therefore an interesting paradigm to trial. And we also looked in quiet, and we also presented noise. So at 5 dB SNR, we didn't make it, we did think about making roving noise, but decided, oh, that's going to be too hard. So we had standard noise coming through the speakers, and we questioned, well, because people with listening deficits or auditory processing disorders have difficulty listening in noise, does the noise change their percept of the localization, right? So that's what we were asking. Now, what did we get? Now, what we did get was there was no difference between quiet and noise, but these are regular young university students who none of them reported any difficulties with their listening. But it would be cool to actually try and see if people who have listening problems, what do they find when you do this? The other thing we found was that actually 30 degrees, when the signal was coming from this 30 degrees right or left, that was the only significant one compared to the 60 and 90. There was no difference here, which is curious because if you're used to doing listening in spatialized noise, actually 90 degrees is approximately where your spatialized noise is coming from in that particular paradigm. But in this population, the 90 and 60 gave a very similar response. And if you look at minimum audible angle, then it's somewhere actually much smaller, close to 10 degrees. That's supposed to be the smallest angle we are able to perceive. Now, we couldn't do that in this sound-treated booth, sound attenuated booth, but that's something worth seeing and trying to evaluate. If there's a difference between 30 and 60, it would be really interesting to see, well, how low can we go with our spatial separation? So that's about the factors that influence P300, and we're close to an hour, so I'm going to, there are only a few more slides to go for clinical studies. I think it's reasonable to start with consideration. The aim here is to understand not just what is not functioning, but it's also to know that can we use these physiological methods to evaluate treatment benefits? And I'm thinking, for instance, people with hearing loss. So once you give, or you change their targets, you change their compression ratio, etc., can we use P300 to evaluate those changes? I'm not sure. It actually hasn't been done. There is one study, and I'm going to talk to you in a minute about it. But there are some considerations we need to bear in mind. First of all, variability in normal subjects, huge. And for all the reasons I cited before, there's so many factors that influence your P300 amplitude and latency. The other thing is, I think this was also cited earlier in that 2014 paper, that we don't really understand the several processes that are related to P300. We know P300 is linked to and links the stimulus to the response, but if you have a small P300 or an absent P300, we can't really be sure what aspect is implicated there. Low specificity. For instance, you can have a small P300 in learning disorder populations, psychosis, alcoholism, schizophrenia. So by no means can we use P300 at this point for diagnosis purposes, or definitely not differential diagnosis. But we can still use it to understand, like I said, the physiological response to changes in cognition, attention, etc. And the last one is kind of most important aspect actually, which is small P300 a cause or effect of a disease process? So for instance, We know P300 requires, the best P300 you get is because somebody pays attention. Now, what if there's a disorder that has nothing to do with P300, however, it's impacting P300 attention, in which case P300 response is going to be impacted because of that. So if that's the case, then that becomes an effect. The cause, of course, would be when P300 is impacted by the disease process. So what we don't know is how to separate these. There is good news. Yeah, sorry.
Sorry, Mrita. I think that's exactly was a question over there, which probably have answered within the last slide anyways. In terms of the intellectual disabilities or cognition problems, will we see such a small P300 response that we won't be able to clinically measure it or clinically sort of evaluate the cause and effect relationship from that particular population? Whether it's recommended to do it clinically or not, that probably is the question in people who have all these problems which are related to cognition rather than hearing itself.
Yeah, so I mean, there's this study, for instance, and I'll come back to that question because it is most important, I guess, from clinical perspective. How do we use P300, if at all, right? So this is a good news study, which is Howie in 2014. What they did was meta-analysis across and this is a fairly recent study. The only problem was they could only get 49 studies, and there were methodological differences. The paradigms were used. Nevertheless, there were two things they were able to extract from that meta-analysis. And in this, they were looking at, can we differentiate the mild cognitive impairment and Alzheimer's disease? And what they did find was that both these disorders show a significantly longer P300 latency. Okay, and the second was that actually even within the mild cognitive impairment and Alzheimer's disease patients showed a much longer latency response. But can I also say that this is in a group study? So for individual data, of course, we need the norms, right? So you kind of have to have your norms, which is why I think in some ways, if you want to get on with the P300 right now, having your client act as your control is possibly a good way to start. So following them up and also family members can act as control, because there are studies that have shown genetically people who are linked, their P300 latency and amplitude seems to be very similar. It has its own, it's not always a clear answer, but nevertheless, the first step is having your own set of norms across ages, and then you can evaluate the person's response at individual base level. Okay. In clinical, yes, P300 has been used in dementia, schizophrenia cases. Schizophrenia cases do show longer latencies. And this is compared to their similar aged or peer controls who don't have schizophrenia. Dementia cases show longer latency, but schizophrenia shows smaller amplitude. P300 has been used for monitoring cognitive functioning in communication impaired individuals. I think that's where this is kind of really useful. And also I would emphasize and say that P300 on its own is not, it's not there yet, but it is a useful technique in a battery. Okay, alcoholism, as I've said, seems to reflect P300 amplitude, as does substance abuse. Childhood disorders, ADHD, absolutely. Now, this is actually an interesting population, because what you can do is, if you come across children who are diagnosed for ADHD, haven't started their medication. Now that's an ideal population where you can try the P300 before or during and follow up, just to see how the P300 changes, and that can be nicely controlled. Now you're not making a diagnostic you're not diagnosing it, but you are looking at, which is what I said at the start, that we're using P300 to look at the impact of treatment. So that's where this would be useful. However, it needs to be an attend-based P300, not an ignore, which means you can't really try this with very young children. Then, as I said, P300 has been used to track effects of medication, and there are studies they've done with ADHD. So pretty much the paradigm I said before, it does help with that. Scalp distribution is also important to observe. So like I said, if you have 2 electrode available in your evoked potential clinical evoked potential system, please use the FZ and PZ. CZ is useful, but if only you have one, then sure, use the CZ. You won't know or be able to differentiate between P3A and B, but I suppose it doesn't matter. P3B is actually more reliable in any case. And if you're not sure, you can always do CZ first and then PZ to see how it's changing. That's another way around that. Autism, P300 has been shown to be smaller and later, but you see this effect more in FZ, so P3A rather than PZ. But again, there's a huge variability in that. Similarly, auditory processing disorders, there've been studies that have shown in APD that basically your P300 is a very small response, but then there've been studies that have shown actually it's not impacted. But then auditory processing disorders are so heterogeneous that it's hard to know anyway. So if you think about P300 as a part of a test battery and you have other measures, including an attention measure, then P300 may be able to add to your paradigm where you can say, do we have evidence of their ability to discriminate between similar sounding sounds, stimuli, etc. So this is kind of a very cool study that was done in 2002 in people with hearing loss. This was one of the first, and actually I really couldn't find another study follow-up by this, but I'm sure there's another one. But in any case, this is something this is kind of a good example of what you can do in your clinics. So what they did was they looked at PZ recording, that's all they did for your P300. Now they did look at cortical obligatory responses as well. They looked at ba and da. So they use speech sounds. They presented it with the loudspeakers so people could wear their hearing aids. I have to say I wasn't entirely sure in their paper if they had used aided versus unaided, but that's something that you've already learned about cortical aided potential, so you can do this. Now what's cool in this is that you can see they use 2 intensity levels, 65 and 80, and they measured the P300 across all these severe to profound, moderate, mild, and within normal limits. And you can see how P300 is basically earlier and bigger for normal hearing, and with the increase in hearing loss, it becomes smaller and later. And profound loss cases basically showed nothing. So I'm not entirely sure that they were actually wearing their devices here, but it's something easy to do and easily that you can follow up with your own, where they can control as your own, they can be their own control. They did note that latencies were less variable than amplitudes, and as the degree of hearing impaired increased, the amplitude decreased. So this is something that's highly replicable, easy, I think easy to implement in your clinic. So what do we need? And this was possibly the question, what is needed for P300 to be clinical ready? I can't emphasize enough the age-appropriate norms. In terms of clarity in determining what P300 we are observing, whether it's A or B, like I said, P3B is more reliable. So if that's your aim, then use the PZ. Okay, CZ would also be able to capture P3A and B. And I think part of the problem in the literature is that we don't have studies that have clarified which is which. P300 at present, and this is me emphasizing again, differential diagnosis, not so much, but we still need to use P300 on how the brain is processing information. And if you can use your own client as their control, best case scenario. That's an easy clinical win. So that pretty much ends most of what I wanted to say, but I did have this one study I wanted to highlight where we wanted to see, do bilingual adults have better attention switching? And what we did was, and I'm going to show you this data, it's a little complex, but what we've done is we looked at DA, and we did it again in presence of noise, yeah, so 10 dB SNR. And so this is what we've done. We've presented the two tasks. One was discrimination and the other was detection. I was really trying to capture both P3A and B. So what we did was, in discrimination, we changed the probability. So the first one, the moment the noise started, we had 60% DA, and I really did not expect a P300 to happen here. But then we had only 10% of the time that DA would have a slightly later occurrence, and then another 10% that it would be really towards at the tail end of the noise. And then we had the detection where we didn't present BA at all, it was just DA. And again, I thought we should be able to get a P300 perhaps in these two, because the probability is so low. So just to give you the timing, and I'm going to show you these arrows throughout in my graphs. So this is where the noise began at 0. 300 milliseconds later, the first DA. 500 milliseconds later, it was 10% of the time DA. And then again, 500 milliseconds later, another DA maybe. And then 500 milliseconds later, the noise started. And then we gave a short gap and then started again. So what did we find? Now, this is at CZ. I did say we shouldn't be doing this, but here, just to make my point, so these are the arrows. They're going to forever remind you where the DAS should be. Now, this is the green, which is we have start of the noise. So, of course, we have the onset. We heard the noise. And then you have the ba, ba, and ba. Is that all right? Everything good?
Yep.
Okay, so then you have the DA, which is only DA, delayed start in the second epoch. And can you see that you have the same response for the onset of noise, but you get this big peak for the second epoch when there's a second DA occurring. And there's nothing else. And then we have the third one where they were discriminating. Sorry, it's a little slow. When they were discriminating DA in that same second position 10% of the time. And again, we get the onset response. And of course, there is a DA 60% of the time, so no surprises that there's something there. So you're looking at the blue graph this time. And what we've got here is a P3. Now, what about at PZ? Now, this is where we expected a P300B to be there, right? So at the first epoch, remember the DA is 60% of the time. I really did not expect a P300. I still have got it, but can you see how big this P300, the second one, is? Now this is again at PZ. This time we have the DA. This is there's only BA here in this catch trial. So actually there's no DA. This should really read BA. Sorry. So you've got the DA at 60%. Yep, we've got something there. And then you have the DA in second positions. It's much bigger. But the question is, do we have a DA in third position? Yes, we do. And these two are very similar. So the arrows here are showing, the red arrows are showing where I see the P300B. What about the detection one. So compared to the first, which is red, versus the second, we think what we've got there is a P2. I did wonder if that was a P3A. So again, I looked at when there was only DA in detection, so there's no BA. So we have no DA, this is only noise. We've got the very similar peak to the first one, and the third one actually looks much different, and I think it's because the time difference is so long. But we don't think we've got a P3A, maybe over here. But this is, we are still processing. I've just collected another participant's data. I'm not sure, but it's curious. And when we looked at PZ, what did we find? There seemed to be this inverted response, which when I looked across the whole head, it seems to be that there's a point of inversion between my cortical and my parietal, which makes me think that maybe this is a P300 response. And actually, I've just realized that I have it upside down. So this is something that in my PowerPoint has done. So this is a P300A. So what does this all mean? My last research, I still don't know. We're figuring it out. Ronnie, who's my colleague, keeps asking me what I think it means. I'm not sure yet. But if you collate all the information we've got so far, P300, it's a positive discriminative potential. We have two subcomponents, an A, which is more frontal central, and B, which is more central parietal. When you're record from FZ and PZ. There are methodological considerations that need to be taken into account. Probability, best to keep it below or close to 20%. Time between stimuli is important. 6 seconds is that magic between your targets. Age considerations, we need the age-related norms. And attention. People have to pay attention to get the P300, a robust P300B. These are all the references. Thank you very much.
That was absolutely fascinating. And, you know, I was jotting down some questions as we went along, and you kept answering them in your subsequent slides.
Oh, okay. That's good.
Yeah, that was very good. That's very well done. If any of the audience members have questions, either you can type them in the question box where I will read them from, or I can turn your mics on one by one and see how we get on. And by the time people are thinking, can I ask you a few things? So first of all, the stimulus that you use, do you use any specific stimuli which is just for the use of your lab, or because my equipment doesn't seem to have the Denver stimulus as such?
Okay, so yes, because I guess we have the luxury of being a research lab, so we can use pretty much anything, right? But if you have a biologic, it allows you to take in a DA waveform, a DA wave file, so long as you keep it below 500 millisecond. That's the trick.
Right.
So you can actually use the same process in the same folder, add other speech stimuli that you want, that you can, you know, they're easily available. You can use environmental sounds. Just make sure your wave file is shorter than 500 millisecond.
Right.
There are other clinical systems, and I am not entirely sure that they allow you to actually include any external tokens, but you know what? They will be able to give you options with tones. So you can use pure tones. 1000 hertz can be your base, often works.
Okay,
and then
brilliant.
You can change it to 1025, 1050, 1100, so on. Yeah.
Okay. One of the participants has asked if you can put the reference slide on while we're talking about questions and answers. Thank you. And the other thing is, how long does it take for you to do it? So once somebody's done, let's say 5 or 10 normals, how long would you expect somebody to put aside a time to do the potentials?
So it is highly dependent on your stimulus duration, of course, and your,
Sure.
1 second, all of that. But I would say anything up to get reasonable results, not including your placing your electrodes, 40 minutes, because you
40 minutes.
want, yeah, you want your deviant to have enough number of averages. So remember,
Right.
to get, you need so P300 is, it's compared to some of the other responses like mismatch negativity, it's really big, right? So if you have about 100 averages, 100 to 150, that's a reasonable average to give you a good P300.
Sure.
But then there's a probability thing that it can
Okay.
only be 10% or 20%. So it does take, keep 40%, 40 minutes. Less is
Sure.
good.
Brilliant. And I'll go to the couple of questions that we have on the question board in a minute. If you have to select one group of clinical population where you would like to do it, I'm fascinated by the fact what you said about the cognitive function testing via P300, where the communication ability is a problem. So if you can't get anything else, then you can do it and perhaps get an idea about the cognitive function. I think that's huge. And I think that is probably one of the things that will develop more than anything else in this regard, because I'm just thinking that they have been around for about 45 years at least in the clinical research domain, and we haven't really adopted them in the clinical domain per se so far, for a reason. And whether the more specialist clinics or clinicians will sort of extend their practice by trying to use it as perhaps we once did ABR, where we didn't have the MRI facilities and we started diagnosing the auditory tumors with ABRs, whether we can diagnose the cognitive functions or dysfunction with the P300.
So diagnosis, I doubt it. However, like I said, it's a good task to look at, for instance, and I haven't done this, so I'm kind of thinking on a fly here, really. But if you wanted to know, so listening effort, cognitive effort has become such a big thing, right? And one of the aspects that's important in those is executive functioning, which seems to be a pretty big component of your P300 system. So I wonder if we can use P300, for instance, for those purposes. Coming back to your point about communication disorders, absolutely, brain injury cases where, they're speech production, aphasia cases, they cannot tell you. You can use P300. We're not diagnosing, but within a test battery, you can start to see how this can be used. So, for instance, I'd come across a case of a glioblastoma. I think that's what it was. So he had to have his right frontal lobe removed, right? And he came out of that with absolutely no appreciation of speech. Okay, his audiogram was mild to moderate. He had a notch, but then he was in industry, etc. We didn't know what he was able to perceive or not. So the one thing we should have done and didn't do could have been the P300. We did the cortical responses. We did the ABRs. ABRs were quite nicely aligned with the PTA. But so that's where P300 can be useful. Again, you're not diagnosing, but as part of the test battery, you can start to see, well, can they differentiate between these sounds? Do they have that capacity?
Yes. And I'm glad you said that, because that was exactly what was in my mind. And I have a patient, an adult patient, who had a head injury, and her audiogram is somewhere like 20-25. And they're very with it, they're a very bright person, and they're in a very good job, and they have claimed for last 4 years so to different professionals that they definitely have speech understanding problem, and they've run several APD tests and all the other tests that you will do for that sort of thing, and they couldn't pinpoint what exactly was the problem. And I wonder whether this sort of task can give objective evaluation of what they're experiencing, and that is very important for a closure for a person when they have a real problem which only started in their adult life, and they've been told from one professional to other that everything is fine, your ABR is okay, your PTA is fairly okay, so you should not have it. Your speech understanding in noise on our tasks is okay. And if you can pinpoint that there is actually an objective problem which we can find, I think that gives a great sense of closure to the individuals, and that works really well for their rehab. And leading on from there, one of the comments is that we do not have a standard test battery for diagnosis of APD. So where we compared the patients where we didn't have APD and we did P300, whether it was a result of a faulty APD testing rather than a problem with the P300 diagnosing it. But do you have any thoughts on that?
Well, so this was the comment I made was a cumulative a result from a number of studies. So yeah, and they're correct that there are so many people use different test batteries. So it's possible that that was indeed the case, but I'm not sure. I think in my own practice, not every person who has come through with listening deficits, for instance, have turned out to have auditory processing. We know that some of them would have attention deficits, but I mean, to them, it's a listening problem and listening in noise. It's up to us to figure it out. So it's possible, you're right, it's possible that that's indeed the case. And we have found, so this last study, the research study that I'm doing, is actually a result of a behavior study we had done with teenagers who had problems listening in noise, and we tested them on every single test that we could think of on auditory processing disorders. It's published by in JSHLR, and what we found there was that the problem was on attention switching, which is P3A. So that's why we're now trying to replicate that behavior study to here. And and if you look at that paper, we only found out of 23, I think there were only 5 who had frequency discrimination problem. The rest had nothing else other than attention switching. So anyway.
Sure. There's another comment here. Should we? I think in my mind can we automate P300 interpretation like we do with the aided corticals and the corticals with the Hear Lab, where you press the button and then in 20 minutes you get an audiogram?
Yeah, I don't see why not. I think NAL is working on some of more on other tasks as well. I'm not sure if P300 is one of them, but maybe yes. I don't see why not. It is such a distinct response to evaluate it from baseline, which is what corticals do from Hear Lab. It shouldn't be a very difficult translational task, but I'm not doing it, so I don't know.
And on the tangent, I guess, I will ask you about the listeners that you mentioned with the localization study, where you said that 30-degree angle seems to be a better angle for testing. Do you think we need some modification in the listeners stimuli today on the basis of that, or are we not as far as that?
See, why change something that works, right? So listens, proven, it works, leave it at that. But what I was pointing out was that if we wanted more, so P300 is able to give us a more, I guess, fine difference in the spatial segregation aspect. So why not? Why not try that? But by no means would that be ready for diagnosis the way Listen has been tested and trialed and tested. So not there yet, but certainly 90 degrees is a massive difference. And if kids can't do that 90 degrees, they certainly won't be able to do the 30 degrees. So I'm just wondering about, I guess, more subtle differences, like your client, for instance, where there's no other problem other than listening in noise. What is their minimal audible angle, and actually, does that matter? I don't know.
Sure.
All we said
Sure.
was that P300 was able to figure that 30 degrees out, and I thought that was very exciting.
Sure. I think the binaural interference is another angle that I'm thinking about them, because
Yep.
they had a unilateral head injury, but that's a topic for another day, I guess.
Yes, after
Um,
I've read that book.
the other thing is, in terms of, you alluded to the fact that you're doing a lot of work in cochlear implantation with the P300 at the moment. So
No, no,
what?
no, not started that, but that's where
Okay.
I'm going with
Okay.
it.
Sorry. Okay. Okay. So are you able to sort of differentiate between? Um, I guess it's a vague and complicated question, but I'll ask it anyways, and you can shut me up. In terms of the presynaptic and postsynaptic neuropathy, where we do not have a really good markers, clinical markers per se, to differentiate where the prognosis of cochlear implant will be better and where it won't be. Do you see any use of these potentials there?
So, yes, it is tangential, that's for sure. But, like Gary Wren's work pointed out about the cortical responses, right? So for P300 to happen, first there needs to be a cortical response. As we know that there is a percentage of of neuropathy cases where we don't see cortical responses. So trying to do P300 there doesn't even, that question doesn't arise, right? But for the remaining, if they're able to attend and we can do a P300, I think this would be exciting to do, because then we can figure out about their discrimination. Having said that, there is a lot of work that's happening with acoustic change complex or mismatch negativity. There's been an interest in this population with hearing loss and in younger children, because to pay attention and do a very simple task for young kids is hard, which is, I think, part of the limitation of the P300 as an uptake for clinical, work. But ACC, the acoustic change complex, and mismatch negativity, actually mostly acoustic change complex, provides us with a more promise, because it is a cortical response. It doesn't require a person to pay attention. They can sit back. It is just a cortical response, but instead of going from silence to something, it's going from one thing to another thing. So it's not a discriminative paradigm, I don't think. We don't know the answer yet, but it's certainly able to tell us a little more about the capacity for discrimination, which is what we want. So theoretically, your question is sensible, because if we are working with children and we figured out that there's an element of neuropathy, then the next question is, well, because that's what we want to know. What is their outcomes, their language outcomes? What are we going to get with their devices? So the next thing we want to know is their ability to not just perceive, but to be able to discriminate. And if we can't do it behaviorally, we need something that we can use objectively. P300 may be a good replacement, or maybe not.
Fair enough. Much as I'd like to go on with this fascinating conversation, I think our time is up. So there are a few more questions which I will direct directly to you, perhaps in an email if the delegates send it to me, or you can just directly contact Mridula if you like. But thank you so much for your time. I know there's a big time difference between UK and Australia, and you're doing it after work. So I'm really, really grateful for your time and fascinating information that you provided us this morning. Thank you.
Thank you. Thank you. I enjoyed that very much. So thank you.
Yes, thank you all. Thank you for joining in. Yes, bye.
Yes, thank you for all the questions. Bye.